Hello, good morning, and welcome to our fabulous FameLab Echoes, which is an event which is extremely exciting for me. I'm hoping you'll start to feel a bit of excitement coming through, because it's a great pleasure. We're going to witness a wonderful series of short talks from international scientists from all over Europe, so you're really in for a treat. And also, what I'm very keen about today is to try and get you involved with some questions. So once the talks are finished and you've got some good questions, some exciting questions, please feel free to start a discussion between all of us here so we can probe our young scientists about their fields. So, how many of you here are familiar with the concept of FameLab in itself? Maybe show of hands. Who knows FameLab? Right. Okay, so not many. That's good. That's great. So you're novices in the FameLab world, which is good. So FameLab is a competition for young scientists, and what usually happens is they have their own national event. In Prague here we have a semi-final for the Czech part of the Czech Republic, and then in Moravia in Zlín we have another semi-final, and the best 5 from each semi-final then compete again in Prague in the grand final to choose a winner who then represents the Czech Republic in an international event at Cheltenham in England, where they all compete against each other. And today we're going to see some of the winners from different countries around Europe who competed at Cheltenham. And usually in all these FameLab competitions, they have a stage like this, and they'll come up and they have 3 minutes. Strictly, there's a timing system with a big gong, 3 minutes to tell you about their science. And afterwards, there's various adjudications being made, and then they're scored, and then at the end they're announced who's going to win. Today, there's going to be no adjudications, no strict timing bells. There's going to be, the only thing that's going to be about fun, and it's going to be an opportunity for you to listen to their work, and an opportunity for you to ask some questions. Okay? Now, I'd like to say some big thank yous at the beginning as well, because we're sitting in this wonderful arena, this room of chandeliers and glass and marble, and that's a big thanks to the lady at the back. If you turn around on the wall, it's Dr. Silvia Pavlicová. So a big thank you to you for able us to come back to this wonderful marble room. And of course, on top of this, that's the place we're going to be, but of course the whole system of FameLab is supported by all these wonderful peoples, where you can see them written in here, whether it be Cheltenham Festivals, Adeto, who supports all sorts of interesting events, Nadace Tomáš Bati. I mentioned that the Moravian semi-final is in Zlín. Well, that's usually held at the Villa, which belonged to Tomáš Bati. Czech centres, the Academy of Sciences, for whom indeed I work, Vodafone, but first and foremost, it's the British Council. And the British Council are involved in all sorts of cultural events here in Prague, and indeed in most cities around the world. And so it's thanks to them that we have this wonderful opportunity to have some fun with science. And a big thank you goes to the lady who coordinates a lot of the work here at British Council for FameLab. That's Mrs. Dáša Seftonová. So thank you very much, Dáša. I think it's safe to say she's become the mother of all the FameLab people and young students who've gone through, and she's doing her very best to make everything happen. So a big thank you to Dáša. But before we start, we've got a special visitor today, indeed from the British Council. She's new to Prague, and I've just been hearing about her history. She's represented the British Council now in all sorts of countries, from North Africa through to Asia, and now we're very fortunate and lucky to have her here in Prague. So, ladies and gentlemen, please a big welcome to the new director of the British Council in Prague, Mrs. Denise Waddingham.
Good morning, everybody. It's a fantastic pleasure to be here in this amazing surroundings for, I think, what's going to be a really exciting morning. You're going to be so impressed with these finalists. The British Council, what do we do? Our work really is in cultural relations, and it's about bringing greater understanding between countries around the world, between the UK and all the many countries where we work. And we work in about 115 countries, something like 260 cities globally. And we do that in a variety of ways. And Dr. Lonsborough's just mentioned we work in cultural events, but it's around exchange, and exchange in higher education and science and research and innovation is really, really important. And, you know, international collaboration in this field is one of the key priorities for the British Council. And I'm delighted, having just recently moved to the Czech Republic, that science communication is so high up on our agenda. And we have this wonderful opportunity to work with fantastic FameLabbers. And the British Council is very fortunate to have the partnership with the Cheltenham International Science Festival in the UK, which was mentioned, and which the winners of FameLab get an opportunity to go to. So I don't want to waste any more of your valuable time. I was just saying earlier that science was really not my thing at school, so I'm a bit ashamed of my my lack of scientific ability. But you know, I think you're probably all way, way, way better than I ever was, and I'm just so impressed by the talented young people you're gonna meet this morning. So thank you very much. I hope you have a great morning.
Thank you very much, Denise. Thank you very much. Well, we all hope that FameLab and other activities are going to open your eyes to science and bathe you with a nice, warm, scientific feeling. So please do enjoy today's talks. Okay, let's begin. And I'd like to begin by asking all of you just to turn around a little and look at your next-door neighbor. Look them in the face. Or turn around to your next door neighbor, you can turn your faces and have a look at them. Study their features on their face. Can you do that for a little bit? Go on, have a proper scientific study of the features. I can see a lot of you looking at me. I want you to look at your next door neighbor and just study them a bit. Study them a bit. What's one of the first things you notice? What's one of the first things you notice? Well, statistically, one of the first things you should have noticed is their eye color. Now, their eye color, and statistically as well, they're probably going to have brown eyes because they're the most abundant amongst eye colors. And that leads me on nicely to our first talker, who is a young scientist from Slovakia, although she has been moving around Europe. She went to Italy and then afterwards to Switzerland, where she's now doing work at the University of Lausanne. And her name is Kateřina Cisárová. If I can ask you to welcome her onto the stage. Kateřina, please join us up here. Welcome, welcome, welcome. So Kateřina, I understand that you're in particularly interested in the eyes. And so I've already asked our audience to gaze into their eyes and to check out their eye color. And I'm hoping you're going to tell us a bit about eye color. But I did take the pleasure of looking up a few interesting facts about our eyes. Did you know that our eyes are our most used muscle of all of them? Of all the muscles, it's our eyes that we use the most. And that indeed, every year we blink almost 5 million times. But I'm going to ask you, can we look each other into the eyes? One thing I noticed about yours, you've got a very similar color to me.
I do.
And I'm hoping you're going to tell me a bit about
that
blue color. Okay, please, Kateřina.
tried getting tickets to Ed Sheeran's concert. Impossible. Gone in 5 seconds. Well, I think I will just pick up the phone, call Ed Sheeran, and say, "Hi, Ed, listen, you and I, we are very distant cousins. So, how about getting me a free ticket?" Yeah, that would just be stupid. Or would it? The thing is, I am related to Ed Sheeran. In a way. It's because all the blue-eyed people in the world, including Ed Sheeran and myself, are descended from a single common ancestor. Oh, and by the way, did you know that while brown eyes are brown because they contain brown pigment, which is called melanin, there is no such thing as blue pigment in human body? Actually, there is almost no pigment at all in blue eyes. And they are blue as a result of light scatter, which is the very same phenomenon that makes the sky appear blue. But let's not get sidetracked. So what was the story of the first blue-eyed ancestor? Originally, all humans had brown eyes. But then, the blue-eyed ancestor appeared with a mutation in their DNA. This mutation drastically reduced levels of melanin in the iris, allowing for the eyes to appear blue. From this single ancestor, the blue-eyed mutation then spread like a wildfire. Nowadays, more than 80% of Northern Europeans and more than half of Czech people have blue eyes. Judging by these numbers, it seems that the blue-eyed people must have been pretty successful at surviving and reproducing. But what was it exactly that gave the blue-eyed people such an advantage in the dark-eyed world? Well, one theory says that the advantage is related to the lack of sunlight in northern cloudy climate. With no pigment in its way, more light can enter into blue eyes, making the dim light vision more accurate. It is also possible that blue eyes spread side by side with pale color of the skin. Light skin can absorb more UV light, which was at the time the only way to get vitamin D, extremely important for bones development and for the immunity. But there is a second, a bit more spicy theory, which suggests that blue eyes spread through a mechanism called sexual selection. You know, sometimes males and females of a species choose their mate based on their attractiveness or uniqueness, rather than their abilities at survival. Just imagine a tail of a peacock. It's magnificent, but otherwise it's kind of useless. And so these are the two theories that we have. But the truth is we don't know why and how exactly did blue eyes spread across Europe. And who knows, maybe one day we will be able to tell if we have blue eyes because of practical reasons or because they are just so irresistible. Thank you.
Thank you, Katarina. So, before I open to questions, can I have a show of hands? Who here has blue eyes? Little baby here at the front. Yes, it really is.
It might be a half.
It really is, almost half of you. So
yeah.
that's amazing how statistics seem to work so well. Well, welcome, brothers and sisters with blue eyes. We are of the same family. Any questions? We can all pick Katarina's knowledge of the eye. It doesn't necessarily have to be about eye color, does it?
No,
we can
no.
ask you. Yes, the young lady here.
Why somebody has green eyes?
Yeah, that is a very good question. So you can think of it like this mutation that appeared was a switch. So it turned off the production of melanin, and this determines if you will have light eyes or dark eyes. And then green, it's a combination of other genes. It basically means that you have slightly more pigment in the iris than the blue-eyed person. So you have green eyes. And gray, that's also gray, it's actually even less than blue eyes, the very pale gray eyes.
Great. Well, can I have a follow-on question? I'm
Yeah.
a great fan of David Bowie.
Okay.
You know, the singer,
Yes.
the musical artist, and he had a strange thing. He had two eyes of two different colors. How does that happen?
So In his case, I think it was an injury, so that could happen. What can also happen is that you have something which is called mosaicism, that basically mutation is active only in one eye, not in the other eye. This could also turn one of your eyes blue and the other one not.
I see, so there's an interesting sort of asymmetry. Okay, any other questions about your eyes? Yes, sir?
Is there any other mechanistic effect of that mutation in the melanin than to have blue eyes? I mean, in the metabolism, something like that?
No. They even checked...
Why it could be, for example, good, this mutation for melanin? Melatonin?
Yeah, it turns down the production of melanin in the iris. And normally, I mean, melanin, we have it in the iris to protect our eyes. That's actually a bit kind of why I chose this topic. I work in eye diseases, and we see eye diseases, some of them are more associated with blue eyes. So it got me wondered how come they evolved, because they don't give us any advantage whatsoever. So it seems they did in past, they don't now.
Aha. So, but does it mean, would as we've got blue eyes,
yes,
do we see things slightly differently than if we had brown eyes?
That was one of the theories, that in past, so where people started to move from Africa towards northern countries, the climate was different and the light was different. It was dim. So this was one of the theories, that our eyes turned blue because we could see better in this light.
That's fantastically interesting, actually. Because I suppose one always assumes that everybody sees everything else as you do yourself.
Yeah.
But then all these things could be rather different. Yes, sir. Another question.
Is the color of the eye and the content of the melanin connect somehow with the quality of your view of your eyes? That's, for example, blue eye guys had better eyes than the...
No, I wouldn't say so, no. No. I have terrible vision. I missed 10 and 10 on both eyes. No connection. With association with the goodness of vision, not that I know of. It would be cool.
Any other questions? Yes, there's a young lady there in the red.
Yeah, I want to ask, because you said that in the past everybody had brown eyes.
Yes.
And now everybody of us, of people, when they are born, they have blue eyes, and later they come to brown.
Yeah, that's a great question. So how it works is a bit like a suntan. So when you have light skin, you expose yourself to sun, you become a bit colored. That's the case with eyes of the baby. So if you don't have the mutation and your gene is working perfectly, your eyes will become dark. But they need to absorb the sun, and the melanin needs to get darker.
Okay, last question, the young lady with the glasses. Sorry, I beg your pardon. My eyesight's also not very good.
It sometimes happens that your inner and outer iris have a different color. How does that happen?
Inner and outer. So as far as I know, you have the inner iris, which has melanin in both cases, in blue, green eyes, gray eyes, except for albinism. If you're albino, then you don't have pigment, not even in the inner iris. And the outer iris, that's the one that has different levels of pigment and is the one that is giving the eye its color. That's why even if you think about albinos, their eyes are pinkish or red, or they can even be slightly violet, and it's because they don't have pigment in the inner layer. And so what you see is the reflection of the vessels in the back of the eye.
Maybe, can I have one last quick question?
Sure.
Do these trends in eye color, do we see them in other organisms other than humans?
We do, but that is not really natural. So dogs, there is a border collie, or I don't know the other species, or huskies. But that's because people bred them that way. People like them having blue eyes, so we kind of did this artificial selection for dogs to have blue eyes.
Because I was thinking that your hypothesis that our eye color changed because we're going to northern climes where there's not so much sunlight, I would expect maybe to see that trend maybe in some other animals as they migrated.
That would make, but animals, if you think about it, they have other senses which are much better than the vision. So I would say they didn't need it. And also for the vitamin D hypothesis, it's different. It's working differently in animals than it did for us. So, yeah. In animals, as far as I know, it's mostly artificial. People selected for it.
Great. Well, can we, another big thanks to Katerina. Thank you very much for coming to
you.
Prague
Thanks.
and telling us about eyes. Thank you very much. Our next speaker has come all the way from Portugal, and his name is David Bidara, and I have to point out that despite in your leaflets there you might have his name spelled with just one R, indeed that's down to the bad eyesight of Deirdre Robertson. He is with a double R, so Bidara is, can I ask him to come to the stage, David Bidara? And welcome to Prague, welcome to Prague. And David, if I'm David or David, or how would you pronounce it?
In Portuguese it's David.
David.
But David is okay.
Okay. And I noticed you also have blue eyes, so
yes, indeed.
We're related closely. I gather when David is not doing his scientific work, he's very fond of playing the violin, if I'm not mistaken.
Yeah, like not a professional level, but just for fun. Yeah.
Great. And what sort of music do you enjoy playing?
I play in a group of music. In Portuguese it's called tuna, but it has nothing to do with the fish. It's academic music groups from the university that play traditional Portuguese songs, and so I'm a member of that group, and we play around the country and sometimes in other countries too.
Lovely. Well, you're studying or working at the University of Porto in Portugal.
I'm just finishing my studies in Porto.
Right. Yeah. And you're concerned with oncology, which, if I'm not mistaken, oncology, of course, is derived from the Greek onkos, which means mass or tumor, volume, and so it's very much linked with the prevention, diagnosis, and understanding of how cancer comes to us in our bodies. And we're very fortunate because David is doing some excellent work regarding searching molecules that are circulating in our bloodstreams, which could perhaps tell us a bit about a developing cancer, if I'm not mistaken. So
yeah, it's used for prognosis. It was a study that we conduct in a group of patients that have prostate cancer. It was like about 200 patients that were submitted to surgery, and then some of them had the cancer again, and some of them did not. And so we are trying to understand why.
So you have an opportunity now, you have a group of people that's giving you an opportunity. So I tell you what, don't tell me, you tell us all about the molecules that you're looking at.
I must say that my story is not concretely about my work, but about cancer in general and a new perspective on cancer, I can say it this way.
Okay.
I hope you enjoy it.
Yes, certainly. David.
In 1859, Charles Darwin proposed a controversial concept that radically changed the way we see ourselves. The concept of natural selection, which says that we are here today because our ancestors won the fight for survival. It was always a matter of fighting. Let's take an example, giraffes. Giraffes with long necks were able to reach the highest branches and obtain food, so they survive longer. And when they reproduce, this trait is passed on to their offspring, while giraffes with short necks were constantly losing the fight and have progressively disappeared. We are the result of millions of years of evolution and natural selection. But here's a very curious thing. If we look at ourselves microscopically, at the cells we are made of, sometimes we see a cell that is stronger than the others, more able to grow and to obtain oxygen and nutrients, that divides more rapidly and generates daughter cells with the same properties. But we don't call it evolution. We call it cancer. Why? Why is it that fight for survival between individuals is a good thing, but internally it can even kill us? Okay, let's see the numbers. We have 100 trillion cells in our body, and most of them are constantly dividing. When a cell divides, all its DNA has to be copied. If each unit of DNA, which is called a nucleotide, is a letter, copying the whole DNA is the same as copying the complete works of Shakespeare 680 times. That's a lot. It's not easy to copy all this without making mistakes. And our cells make about 120,000 mistakes, which is a high number. Many of these mistakes can give the cell those abnormal growth advantages and form a cancer. We could even ask, so why don't we have cancer every day? Well, the fact is that our cells try really hard to stop this evolution process. First, they are able to read the DNA and to correct the mistakes they found. More than 99% of mistakes are corrected this way, but some of them may escape. It turns out that a cell that is created with errors, and so potentially dangerous, has the extraordinary capacity of committing suicide to avoid an abnormal growth. This phenomenon is called apoptosis, and is always happening. Since I started this talk, more than 100 million of your cells have died from apoptosis. But let's think about all this. It was the fight for survival and natural selection that allowed us to be here today. But it's the sense of community and coexistence of cells that keeps us alive and healthy. Thank you.
Thank you, Dave. That's a fascinating sort of almost paradox of that life is guaranteed by our own sort of cellular suicide. Does anybody have a question about your cells for David and their evolution?
Can I just have a bit of water, please? Thank you. I'm really thirsty.
Any questions here? So, well, tell us then a bit more about how when a cell is committing cell suicide, or if it has got an abnormality about its growth, does it then release certain molecules that we can detect? How quickly can we understand that something is going awry?
Cells are committing suicide all the time. So the problem is when they don't, when they are able to overcome this situation and not to kill themselves. Because when a cell is abnormal, the own cell starts to, okay, I have to die because something is wrong in my DNA. And when the cell doesn't do that, the other cells tell her, oh, you have to die because our DNA is very dangerous. But sometimes the DNA is so dangerous that the cell is able to ignore all the advice and still growing, and that's a cancer.
So it's fascinating how you talk about it, because it almost puts into my mind an idea, concept that there's a decision maker in that cell. Who's making the decisions? Who's actually saying, there's something wrong here, let's kill it?
I don't have an answer for you.
Yeah. And so it's some sort of protein that's reading the DNA and then making decision?
Mm-hmm.
Okay.
We have a very strong machinery, I guess, of reading the DNA and correcting mistakes, because the proteins that make DNA replication, they commit a lot of mistakes, about 100,000 mistakes for each division, which is a lot. And we have to correct all of them, and most of them are corrected, it's good, but some may escape, and the mistakes that escape, they are the problem.
Okay, and are they always a problem, or are they sometimes an advantage? I suppose that's the evolutionary key, right?
That's evolution, yeah. In a long-term perspective, they can be a good thing, and we are here because of that, because of those tiny little mistakes that allow from a single cell and the first bacteria to us. And so we are, we directly descend from that kind of mistakes, but sometimes the mistake is so strong that it can kill us, and that's a cancer.
Yes, the young lady here at the front.
Thank you. My question is, the process of killing or sort of stopping the cancer to grow is down to the other cell's ability, to your body ability to do that process. But my question is, if you are tired, not fed well, exhausted, whatever, stressed, are these conditions helping the cancer cells to survive, to escape that controlling system?
It's very controversial, actually, but it can be true. We are not sure, but everything that belongs to our daily life can help precancerous cells to grow and become a cancer, starting by the food we eat or the time we sleep or our stress. because stress is directly connected to inflammation, and inflammation may trigger cancer. And so everything can help cancer cells to become more dangerous, or by contrast, we can also make them less dangerous and make them die. And that's the good thing. That's why we should eat healthy food with a lot of antioxidants, because they can help precancer cells to become smaller and not to grow up, not to become a cancer. But there are a lot of variables, because it's not a genetic thing, actually. It means, oh, we are, the destiny told us that we are going to have cancer. It's not true. Actually, our routine in our daily life can have a big impact.
But there is to a certain degree, I beg your pardon, yes.
So it's possible that I had cancer, but it killed itself?
Yes. Actually, you have it every day. It's not, we use the word cancer when a cell is very, very dangerous. We use the term tumor. That is a group of cells, of malignant cells that have this type of behavior, but they arise from very small populations of cells, which are always trying to grow up, and the system is telling them to grow down. And so if this happens, if the system is doing okay, we are free, so there's no problem. So don't worry, no problem with you.
Two gentlemen there.
When we long time on the sun, we get burned because the cells are committing suicide. So why are we getting freckles sometimes?
Getting? I didn't understand.
Freckles, spots, the dark spots.
On the skin?
Yeah.
That's because the sunlight can cause direct mutations on DNA. And so they are very dangerous mutations because they generally are irreversible. The body sometimes cannot correct those mistakes. And so that's why sunlight, especially the most aggressive rays, gamma rays and so on, ultraviolet. I'm not an expert on that area, but they are very dangerous because they can cause mutations that our body cannot correct.
I don't know if I understand this correctly, but from what I've heard, the cells are created out of chromosomes, and chromosomes decay over time. But the chromosome, and that's why you grow old, but the chromosomes that create cancer cells do not deteriorate with time. So theoretically, if we could coexist with cancer cells, we could live indefinitely, practically.
I'm not sure, because the problem with cancer is not only at the chromosomal level, but at the gene level. Because cancer cells have a lot of abnormalities in the number of chromosomes, but the first abnormalities to arise are at the gene level. And so just because cells exist, they have to replicate their DNA. And just because they have to replicate their DNA, they may commit mistakes. And each one of these mistakes, if it's not corrected, can generate cells that become more and more powerful. And then some abnormalities at chromosomal level start happening. But they are not, usually, they are not the first, the more relevant happening. It's at a smaller level, at gene level. I don't know if I answered your question.
Yeah, I think you, yes, young lady.
You said that cancer is not a destiny. Does that mean that when our parents or grandparents have cancer, that we have a bigger chance of having it too, or not?
Yes, actually, there is a component, a familiar component, how can I say? It depends on the type of cancer, but about 10 to 15% of cancers, I guess, may have a familiar origin, which means, for example, prostate cancer. My risk of having prostate cancer is very low because I'm still too young. But if I have a father or a brother with prostate cancer, I have the double of the risk. So although there is a familial component, it's not everything. It represents actually a very small percentage of cases. The majority of cases are caused by the environment, I guess. So our life, the food we eat, etc. Those examples I was giving to you, they are the big factors, I guess, because it's not a destiny. I mean, if my grandmother have cancer, I will have cancer too. It's not necessarily. It's just a small component.
Okay, David, I'm gonna submit. Thank you. That was a fascinating discussion to hear about this sort of competition between cells, between chromosomes, between individual genes, and that sort of within the context of that very competitive world, we have to deal with these problems cellularly. So, thank you very much indeed, David. Thank you. Welcome.
Thank you.
Our next speaker is our local hero, the young and beautiful Tereza MAŠÍNOVÁ, who indeed won the competition here in the Czech Republic. Congratulations this year. And she's here again to tell us about some of the work she's been doing. But apart from being a scientist, you're also a keen tap dancer.
Yes,
if I'm correct.
that's true. Yeah.
So, I mean, I suppose this is the wrong sort of surface to be doing some tap dancing, but perhaps on another occasion you can show us a bit of your moves. But yes, today you're going to be speaking. Your specialization is in the environment, but not necessarily an environment we can see so well. It's the microbial environment, isn't it?
It's true.
So it's the environment around us which is more or less invisible to our eyes, which reminds me of a quotation from a philosopher and indeed a scientist, Goethe. And he said famously that you see what you know. And therefore that the idea that what we see, that our vision is not just a function necessarily of what we can really just see, but it's a function of our intelligence, a function of our imagination. And we're very lucky today, ladies and gentlemen, to hear from Tereza, who's going to tell us a bit about what she can see in terms of the invisible microbial world. So, looking forward to it.
Thank you. Thank you. Hello, everybody. Do we know each other? Let me introduce myself. I am Prochlorococcus, unicellular cyanobacteria and an important part of the marine phytoplankton. I belong to the smallest photosynthetic organisms. You just caught me doing what I'm best known for, photosynthesis and its associated oxygen production. Did you know that we, members of the Prochlorococcus family, produce about 20% of all the oxygen that is released on Earth during photosynthesis? Okay, I know that tropical rainforests produce twice as much as we do, but you have to take into account that unlike rainforests, you cannot see us with the naked eye. This makes our achievement even more remarkable, am I right? You might be wondering, how is it possible that something so small as us is so important? The answer is simple. There are quite a lot of us. About 10 to the power of 27 individuals. And yes, we are the most numerous species on Earth. Did you know that we occupy the same volume here as you, the human race? I know what you are thinking. How do we do it? First, we live in tropical and subtropical waters of world ocean. These waters are known for their low nutrient content and thus the other species can't survive here. Therefore, there is a very little competition. 2. Low nutrient content does not matter to us. Our minuteness actually gives us a clear advantage in receiving nutrients. We small spheres, unlike the big spheres, have a large surface-to-volume ratio and therefore this little amount of nutrients easily permeate into us. And 3.
We multiply very quickly. We double in number every day. At such speed, by the end of this month, I would have more than 1 billion descendants. Half of us are lost along the way to thankless predators, but we are still the most numerous species on Earth. So remember, the fact that you cannot see us doesn't mean that we are not essential to you. Did you know that every breath you take contains oxygen that was produced by me? Thank you.
Any questions to the organism who gives us oxygen? The most numerous, yes, sir.
Is there idea to cultivate you for the producing of the oxygens manufacturally, not naturally?
Of course, there is many attempts to do so, and I think that people are very close to being successful. So it seems that I might be used in the future for production of biofuels or something. Yeah, so yes.
So do they think that it could somehow be used instead of the oxygen from the tropical forests which are cut it down? And
it's a very interesting idea. I've never heard about it. But I will try to suggest it to some researchers.
Who produces more oxygen for us? You or the rainforest, for example?
Rainforests in total. But their biomass is also much larger than my biomass. So
you're more efficient?
Yes.
Any questions for, yes?
Are you, or how good are you in being stable and, resistant to the radioactivity, to this kind of generally dangerous radiation?
Well, I would say that I can cope with it pretty well, because I don't need very many things to survive. I just need a bit of carbon dioxide, I just need a bit of water and so on. The question is how my DNA would deal with it. So
in the case of the disaster, nuclear or
well, I would say that I would cope with it much better than you humans.
Okay.
That's for sure.
So potentially you could be used to terraform a far-out planet or moon to change its atmosphere?
Yeah, let's try. Yeah.
Very good. So you've got not just an international presence, but an interplanetary potential. Any more questions for our microorganism who produces oxygen? Or indeed any other microflora or fauna? There was an oxygen deficiency there, somebody yawned. No. So, well, what other species are you interested just in this species, or do you stick to yourself, or do you like to investigate other microbes?
Like...
you personally, now I mean the human you.
The human me, okay. Well, I don't work with these organisms. I work with some other that are living in forests, but I find this one very interesting, and that's why I choose to tell you a bit more about it, because I find it to be really important to know about it, because it's really important and people don't know about it very much.
So if I'm going for a walk in the Czech Republic, a forest, say, and I just take a random handful of soil, how many, can you tell me a bit about what is there that I can't see?
Well, there is like the whole space of microorganisms. There are billions of bacteria, thousands of protists, and actually hundreds of meters of fungal mycelia that you just can't see, and they are there. So it's totally unexplored world that we don't realize surrounds us.
And are many of these different strains of bacteria, are they literally unexplored? Science still hasn't revealed what they could or could not be useful?
Yes, that's for sure. Did you know that we can cultivate about just 1% of microorganisms on the Petri dishes? So the rest remains unexplored, and we have no idea what these organisms are able to do, what's their role in the environment, and so on. So I guess that, or this could be like a very interesting direction of research in the future years.
And any other questions about? Because I had the little babies left, but I remember hearing from several. I've got three. Well, I've had three little babies, and I remember hearing on several occasions that it's quite good to let your baby go around the garden and get some soil and put it in your. You shouldn't stop it from putting its fingers in its mouth, and so there's an interaction, and it's been interacting with some of these microbes. Is that a good thing, or would you not recommend it?
Well, I think it's a definitely good thing. There is actually a hypothesis called hygiene hypothesis that says that people have these allergies and civilization illnesses just because we are not surrounded with microorganisms, that our immune system is kind of bored so that he attacks things that are normal for us, and that's why we have these allergies. So, yeah.
Ladies and gentlemen, Tereza MAŠÍNOVÁ, thank you very much, Tereza. Our next speaker has come all the way from the Emerald Isle of Ireland. Indeed, was the winner of this year's FameLab in Ireland. Mrs. or Doctor, I'm not sure quite of her. Is she a doctor or a master or an engineer? Deirdre Robertson, please take the stage with me in a warm welcome, please. Wow. You're looking very sporty, and you're looking very sporty indeed. Now, I'm a big believer in mind over matter, and one of my favorite poems. There's if I could just read a little bit out for you. There's a section where it says, "If you can force your heart and your nerve and sinew to serve your turn long after they are done, and so hold on when there is nothing in you except the will that says, hold on." So it's about a mind over matter sort of control. This idea that our brains, our minds can push us to sort of physical activity, which otherwise the rest of our body is saying, "I've had enough." Now, you of course are studying a lot about what the brain can do, how the brain works, some of these interactions. So I'm thoroughly fascinated I'm waiting with amazement to see what you're going to tell us a bit how tennis is interlinked with some of these mental processes in the central nervous system.
Yep.
The floor's yours.
Okay. Okay, I can't play tennis. If I tried to play tennis, I'd probably trip over my own 2 feet and knock myself out with this racket. But what I can do is explain why understanding my terrible tennis movements can help us to develop treatments for some of the symptoms of Parkinson's disease. So if you don't know Parkinson's disease, it's a movement disorder, and you might have seen it. It's most commonly characterized by a shake or a tremor. But I'm going to talk about a different symptom. So I want you to imagine this. You're standing at your front door, ready to go in, when suddenly you can't move. Your feet are glued to the floor. Freezing is one of the symptoms of Parkinson's disease. It can happen without warning, and we didn't know why or how to fix it until scientists discovered a fascinating way to unfreeze the brain. So the part of the brain that controls movement is the motor cortex, up here. And it controls 2 types of movement, one which is focused on goals and one which is focused on automatic movements like walking. So it's like learning to drive a car. If any of you have done this, you might remember how difficult it was to remember everything at the start. Steering wheel, brake, accelerator, changing the gear. But now or after a while, it just becomes automatic. You don't think about it anymore. Now, when I picked up this tennis racket, my brain switched on the program responsible for goal-focused movements. It received a ton of information about where to put my hands and where to put my feet. But let's say instead we gave this racket to the famous tennis player Venus Williams. Because she has done this movement thousands of times, her brain no longer needs all of that information. It just switches on the autopilot program, meaning that she could effortlessly continue this conversation while probably smashing the ball into all of these beautiful chandeliers and smashing them over you. I won't do that, I promise. Okay. So what does this have to do with freezing and Parkinson's disease? Well, the brain can sometimes act like a computer, with one program frozen and the other one working. In Parkinson's disease, the part of the brain that starts to break down is that responsible for automatic movements, like walking. So what the scientists discovered was that if they drew a line on the ground or rolled a ball in front of the person who was frozen, they would be able to start walking again. And that's because the line acts as a visual goal for the brain to focus on. That's essentially like pressing Control-Alt-Delete on your frozen computer. It tells the brain to ignore the frozen autopilot program and to switch back on the program for goal-focused movements, meaning the person can start to walk. So it's research like this that shows that by examining the complex networks of the human brain, we can try to develop new treatments for those symptoms of Parkinson's disease that don't respond to medication. And that's because the brain sometimes acts like a game of doubles tennis. You always have one player on the back line ready to take over if the player at the net misses their shot. Thank you. I was wondering where that went.
Fascinating talk, fascinating talk. So, any questions about muscle memories? I've just started, I'm a big fan of cricket. I've just started a little cricket club at my local town for the kids. And we've been talking to them about muscle memory. Because at the beginning it's very difficult to think about getting in line with the ball, where you should be, what parts of your body should be where. And you begin to realize, as you said, that huge amount of information which they have to work out, and then it becomes, I'm trying to convince them to believe that soon it will just come automatically. But it's fascinating to think that if you're frozen, if you change that you switch to a goal-oriented sort of thinking, why do they lose the ability to do that themselves? Why do they need the stimulus?
Because, there's a part right in the center of your brain called the basal ganglia, and you might have heard as well of the chemical dopamine, which is what is, a real problem in Parkinson's disease. Our brain loses the ability to use dopamine. And in the basal ganglia, when, exactly as you're talking about, when a movement becomes automatic, the strong activation for an automatic movement is in a different part of the basal ganglia to when you're starting to learn a movement, when there's more input from the front of your brain and from other parts of the brain as well. So essentially what it's doing is just using the still working part of the basal ganglia. It's essentially like learning to walk again. So it's as if you'd forgotten the ability to play cricket and you're having to learn it again. And that's what it's doing, is just switching to a different pathway to get around the problem. Now, it's not a treatment for Parkinson's disease, and it can still happen. It's kind of a workaround. And there isn't a treatment for Parkinson's disease yet. But it's just a way of using, because we've lots of different pathways in our brain, kind of like if you imagine a metro map, and you can get to one location using lots of different routes, and so it's like using a different route to get to that same action.
Fantastically. Any questions about your brains? Have you ever had a question about how your brain... Yes, please, sir.
Is it possible that your goal-oriented thinking freezes while the automatic works normally?
Yeah, so in Parkinson's disease, now the other thing is that in science, anything can change as well as we discover more things. At the moment, it seems to be that it's the automatic movements that are most affected, and that's why it's things like walking are a problem, because that's something that you learn when you're, 9 months to 1 year, and then when you stop thinking about it, it's an automatic movement, and that seems to be what happens in Parkinson's disease. I don't know whether the goal-focused movements can freeze, leaving the automatic ones. Sleepwalking, sorry, I was talking about in Parkinson's disease specifically. Yeah, that's possible in sleepwalking, I suppose, because you're just, doing the movement without being aware of it.
How about, does any of this relate to being scared stiff? Because when you started mentioning about, of course, in relation to Parkinson's, one of the biggest symptoms is that you suddenly freeze. I was thinking in my life, I can remember just once where I literally was frozen, and that was because when I was a young boy, I used to be petrified of spiders, and we were walking in the Lake District. I was about 7, and my dad was in the path. I can remember my dad was walking in the path. I was walking along the heather, and I stepped in this patch of moss, And from this moss suddenly came, it must have been a spider nest, because suddenly all these spiders sort of came, hundreds of them started coming up my leg, and I couldn't move. I literally could not move. And I had to, I was telling my dad to come up and get me, and he was saying, just go from it. And I said, I can't, I couldn't move.
Wow.
So I was wondering, is there anything, are these things at all, related?
Yeah. Well, I don't know about that specifically. It's never happened to me that I've been completely frozen, but that's probably to do with the stress response where you're, when you're in flight or flight mode, I don't know which you were in at that stage, but if your heart was pounding so fast and all the blood was rushing to your muscles, it was probably more a psychological sensation of being unable to move rather than physically being unable to move.
Of course, it must be a...
Yeah, so it's,
But so where is that, but then is it always not the case if it's when does it become you're physically unable to, in the case of Parkinson's? I mean, presumably it is always, it's a psychological effect.
No, in Parkinson's disease, it's certainly to do with the loss of the chemical of dopamine.
Right.
So it's not it's not the case that if you tell someone, oh, just think about walking, who has Parkinson's disease, they can walk. They can't. This is a way to try and essentially hack into the brain. With other types of feeling frozen when there's nothing essentially wrong in the brain, then it probably is more of an emotional or a psychological reaction. So I'm not saying that if you just said to somebody, oh, start walking, stop shaking, they'd be able to do that. That wouldn't be the case. But there are lots now of companies which have tried to develop treatments or like ways for people with Parkinson's disease to use lines in their daily life to keep them doing. So instead of having to have someone draw a line in front of you or have a ball in front of you, to have like laser pointers on their shoes, for example, or if you have a walking stick, to have a laser pointer on the walking stick which acts as a constant visual reminder of where you should be going. Or at the moment as well, there's companies developing beats, so you listen to a metronome beat in your head because it can be activated by the audio system as well. So if you just hear kind of tick, 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 that acts as a visual goal as well. So to have a constant reminder as well.
That's fascinating.
To help people work around those symptoms.
It's fascinating. So it's fascinating. Yes.
I want to ask, where in our brains are saved this information we have done and we forget? Because for example, I play guitar and I play sonatas or etudes or something like that. And after a few years, I forget it all. And after a time, I try to play something, and I play a short part, and it clicks, and I can play it all.
Yeah.
So where in the brain it is safe, or is this how is this limited?
Yeah. So it's a network as well. So your hippocampus is the memory storage part of your brain. And it used to be, when we understood the brain, that we thought there was very specific little parts of the brain that did certain things. So memories were stored in the hippocampus. And when you tried to think of something complicated, it was just using your frontal lobe. Whereas now we understand that it's pathways. And the more you do something, the more the pathways become strengthened. So that's what happens as a baby when you learn that these movements are walking. The more you do that, the more it becomes strengthened. And that means the pathways just can activate quicker. But if you stop doing something, then essentially it's like it's not as efficient anymore. So the memory is still there in your hippocampus, but it's not brought to mind as easily because you have stopped doing it. So it's similar. You have, when you're playing guitar, you have input probably from your hippocampus, from your auditory system, from your frontal system, for thinking about it, from your motor cortex, and it's just about making that pathway really efficient and then, continuing to use it.
How is it limited? Or it's infinite space? Or
Well, actually, only this year, there's the first research to try and figure out exactly how memories are stored, but it's still pretty complex. It doesn't seem to be that, it's not like having a memory card in a camera, for example, where you have a little space where your memory is. It does seem to be that it's essentially like electrical wires, and when you press a light, the same electrical wires go on to make that light shine, and it's those series of pathways, kind of the best way I can describe it. But it's a fascinating area, and there's a guy called Thomas Ryan in Trinity College Dublin who has just started to look at really what are memories as the most basic question.
So is it true that we actually don't forget anything, that we remember, that our brain remembers everything, it's only a matter of time and how deep we have to dig to get it back?
No, we do forget things. A lot of the time, so when you try to remember something, or when you're, first of all, trying to learn something to remember it, there are two parts of that. So there's the encoding, which is the part where you're trying to remember it, and then there's the retrieval, which is where you can actually remember it. So there can be a problem in either part. So with something like dementia, for example, you might have problems with retrieval, where you can't remember something you've already learned, or you might have problems with encoding, where you never actually learned it in the first place. So there's loads of really clever neuropsychological tests that try to figure out where somebody is having the problem, whether they never learned it in the first place deeply enough in long-term memory, or whether they're having problems retrieving that. So I think the more things you learn and the more experiences you have, it depends how you describe forgetting, but it's probably a problem with retrieval, all right. But how you could access it, I'm not sure. I don't think it's like you can have some hack to suddenly remember everything that's happened to you in your life. But certainly, everything we learn, the basis of all human experience is learning how things are associated. So if you've heard of Pavlov's dogs, for example, the bell was rung and they started to salivate. And that's the basic type of learning in our brain, and everything, our whole personalities and the way we see the world and the way we respond to the world is all made up of those fundamental building blocks of learning. So if a memory is, understanding how two things are associated without explicitly remembering when I was 2, this happened to me and that's why I have this association, then yes, I guess we do remember everything. So I suppose remembering and memory are such a complex, concept. Depends on the part.
When are we, for example, sitting and staring at one point and we ignore the others, we don't listen to them, and we are only looking at the one point? Is it a little kind of Parkinson?
When you're actively staring at something?
Yeah, when we, for example, it takes, I don't know, 20 seconds or something like that, but we ignore everything but staring at one point.
That's, to a certain extent, that's kind of your conscious decision. Do you mean when you kind of, you let your eyes blur and you just want to think about nothing except that one point? That's just a conscious decision that you're making to focus all of your attention on one point. So if, for example, something happened and somebody clapped behind your ear when you weren't expecting it, you'd be able to snap out of it and you'd be able to look over your shoulder. So that is something consciously that you're doing rather than a problem. And, well, unless, it depends on
it's
how
an
you
interesting
come to
thing,
that state.
I've started doing some meditation in the evenings.
Yeah.
And indeed, I try to do that. I try to actually achieve that state, and which I find incredibly difficult to sort of almost wipe out any other conscious thoughts other than this sort of one thing. But so it's interesting to hear that question, that it might be, there's obviously a spectrum of ability to be able to get, consumed in just this one focal point.
Yeah, difficult to do.
Any other questions? Yes, young lady.
I'm not sure if this was already mentioned, but as part of Parkinson's disease, that moment when your part of your brain freezes over automatically, what causes the freezing? What is the symptom? What is the trigger to that?
Yeah, well, the trigger is actually really interesting, and it's not really understood. So, for example, the most common places, the reason I use the example of going into your front door is when you're in a narrow space. And that's not really known why that's the case, that something like a doorway, so external, would cause this reaction. It's a really unpredictable disease as well, so only about 50% of people, well, not only, it's a big proportion, but only about 50% get those freezing symptoms. And the initial symptoms of Parkinson's disease can be really different for different people. So it's pretty unpredictable when certain things happen, what the progression of it is. And we're still trying to figure out exactly what is happening. But it seems to be a problem with, you've different parts of your brain that allow movement and different parts that inhibit movement, and it seems to be a problem with how they work together. So I can lift my arm up and stop it here, and that's a voluntary movement, but if I had Parkinson's disease, this movement might happen involuntarily, and I won't be able to stop it, and that's because the part of my brain that's inhibiting the movement isn't working. But then the freezing is the opposite. It's where it's the part of the brain that initiates the movement is no longer working, so you can have both symptoms at the same time, so it's quite unpredictable.
And do you know what causes, that your brain does either that or that?
No, it's all dopamine related, but as for the kind of specific moment at which somebody freezes or somebody has a tremor, as far as I'm aware anyway, it's still kind of trying to figure out exactly what it was. And if we knew exactly what caused that, then you'd be able to develop treatments to cure it.
Is the dopamine not being released, or just not being caught onto by the cell?
The cells that produce dopamine are starting to die in different parts of the brain.
Okay. Well, I'm going to ask you to voluntarily put your hands together and give a round of applause, please, to our talker. Fantastic. Thank you very much. Our penultimate speaker of today's event comes from Italy, but he indeed has been working for the last, I think, 7 years, he mentioned, in the Netherlands. And indeed, he's the winner of the Netherlands FameLab competition. Please welcome Roberto Nassisi.
Very good.
Buongiorno.
Buongiorno.
Yes, so I'm going to. Of course, you're interested in stem cells, if I'm not mistaken.
Yes.
And I can remember my mum telling me when I was a little boy. She said to me, Michael, when you grow up, you can be anything. Which, of course, you know, that sort of set that parallel with the stem cell. And as I was getting older and older, then my ability to do whatever is becoming more and more narrow. And eventually I'll only have a single purpose.
You still like stem cells. So more they develop, more what they can do.
Exactly. Exactly. That's my. I was going to continue with my parallel that really our own ability to sort of take advantage of opportunities narrows with age, and indeed that's the case with stem cells. So you know, I'm very much looking forward to hearing your talk, because from that, logically, you can conclude that stem cells have an enormous potential to heal and regenerate and open up new opportunities.
Yeah.
So
and they naturally do that every day in our body. So.
Well, I'm hoping you're going to tell us a bit about that.
Yeah.
Okay.
Thank you. We have all had at least one moment in our lives when a big change happened. Something that twisted our way to see and feel the world around us. A moment in which we made such an important decision that from that day, nothing was the same anymore. And we felt really good. Well, I can tell you special cells in our body are making this decision continuously, also in this moment. I'm talking about stem cells. Stem cells are something incredible. They are amazing cells we all have, and they are there, waiting to do their best, change their lives, and reinvent themselves to become something else. This is exactly what's happened during the regeneration of our tissues, like our bones or our skin. However, not all stem cells are the same, and like us, they have their own preference for what they want to be when they grow up. What I'm studying are the characteristics a stem cells needs to have to preferably become a cell of one specific tissue. It's a very, very important tissue that allows us to move freely and smoothly. It's a tissue that, when damaged, causes chronic pain. And I'm talking about hundreds of millions of people in the world. This tissue is called cartilage, and what I'm doing is finding the best stem cell I can use to repair cartilage when it's damaged. And I'm particularly interested in one specific kind of cartilage, the cartilage that we have in our joint. So the tissue that covers our bones at joint level, like in our knees or in our hip. So how can I select the best stem cells for cartilage? Well, normally scientists looking at certain characteristics on the surface of the cells. However, never judge a book by its cover. So imagine, for example, that these are two stem cells. What I found is that sometimes they have identical surfaces, but a very, very different content. And now, with the new technology available, I can mark and select the stem cells for the content I want, which is indicative for the presence of one specific factor that I found to be crucial to regulate the twist of a stem cell into a cartilage cell. And guess what? The name of this factor is twist. The best way I can describe you the magic here is that stem cells that express a lot of this factor, a lot of this twist, are also the best at making cartilage. So stem cells are working really hard every day in our body to make all these decisions. And it's I think, incredibly important to know all the little secrets of these decision-making processes, to be able to master them, treat our diseases, and make us feel good again. Thank you. So tennis is becoming popular, eh?
Yeah. Tennis obviously is the sport of the day. So there's stem cells and stem cells. That
Yes.
means some have a preference. They're not necessarily can be absolutely anything. At least they have a preference to be something rather than something else.
Yeah, the point is that, by definition, they can do wherever they want, okay? When at the really beginning. So when in the first day of our life, we generate a kind of stem cell that actually give rise to all the tissue that we have in our body. So that first stem cells that we have, in a way, can be really wherever they want. But more we go on and we develop, more stem cells that we have in the body have, let's say, some kind of preference. But we have a kind of pool, a reservoir of stem cells in each of our tissues. And this is very, very important to do what? To compensate all the apoptosis that we have every day. Because if you don't replace all the cells that undergo to apoptosis, we are gone in a few months.
Yes.
Really.
So yes, and that's another question I had for you, because you're sort of hinting to a difference between sort of fetal stem cells and stem cells that even an old fogey like I have still in me. And which are more useful for scientists?
Really difficult to say, depend on the perspective and what you are looking at. I'm not a developmental biologist, so I'm mainly working on, we call it adult stem cells. So the stem cells that are already addressed in a certain lineage, that they have some kind of preference. So the cells that we can find in our tissues. A brilliant source of stem cells that we have is our fat. For example, we can get a lot of stem cells from that. They can do still a lot of stuff. So they are quite interesting because there is a dual benefit. So you remove a bit of fat and you can maybe repair even your tissue afterwards if you need it.
Aha.
Yeah, but the developmental biology is really, really fascinating. And without that, we couldn't know a lot of the stuff that we know also of the other cells in our body.
Fascinating subject. Is anybody a question about your own stem cells or other people's stem cells, what the potential of stem cells could be? Yes?
I've heard a theory that stem cells can be used to regrow lost limbs. Do you think it is possible?
Yeah, it's not really a theory. It's kind of true in certain kind of animals, certain kind of lizard animals. And that is a very, very, very interesting model that a lot of researchers are trying to investigate, because indeed, as you said, there are certain kind of animals that you can cut a limb, they can regrow. And try to understand which are the mechanisms that are going on there is actually taking a lot of researchers busy, and seems that there is a lot of that the immune system is playing a big role, so different kind of cells in the immune system can signal the other cells and tell what they have to do or not. We are a bit far still to really translate these and make a human limb regrow, but it's a really fascinating theory, and there is a big bench of research focused on that.
Any other questions from the audience? Well, so it might be a while away to regrow a new limb, but speaking directly, you're concentrating on the knees, the cartilage around the knees, which is good news for me, because I've always enjoyed my sport, but I've always been quite a big, heavy guy, and so I'm starting now to get the first sort of signs of pain in my knees. Yeah, so I'm looking forward to success.
Yeah.
So the question I have for you is how likely is it that I'm going to be able to inject some stem cells and...
I'm afraid to say that in your situation it's a bit less easy. I'm sorry for that, because if you are progressively feel more and more, it's possible that the problem of your cartilage is more a degenerative process and is not a focal defect due to a trauma, for example. And if something chronic is going on, it's not only the cartilage, but all the other tissue around that are not behaving really well, and it's a bit more difficult to tackle all the different components that we have.
The whole thing.
So, but I mean, I have some hope. I think that we are going in the right direction. There are brilliant researchers that are really focusing on, for example, osteoarthritis, which is one of the most common chronic disease of the cartilage, and. I mean, if I have to give you a number, I think that in probably around 10, 15 years, we will have something really good also for that kind of chronic diseases.
So roughly along the same times when nuclear fusion is going to get online?
Yeah, they say so, right?
Probable.
Yeah.
Yes, who's looking forward to tapping into stem cell science to improve their own physique and health? Anybody here is anybody here who's rather skeptical about the use of stem cells and stem cell research? Is anybody here who's against the idea or the principle of it?
I'm not skeptical, but I'm very pleased that I learned that a little bit of fat is good.
Yeah,
yes.
Yeah, absolutely, in a way. And even the more skinny person in the world has at least 12–13% of the body weight of fat, so...
And that's enough.
Absolutely.
Well, on that fatty note, I'm going to say thank you very much indeed for your contributions. Fascinating. Arrivederci. Our final speaker is also from the Czech Republic. She was the runner-up of our local Czech competition in 2016. Since then, she has recently also become the runner-up in a science flash competition in Latvia. She's an amazing individual who has all sorts of interests, is an author of many books and texts. Please welcome Julia Nováková with me on the stage.
Thank you, Michael.
Hello, Julia. Julia does some really. I've been reading some of your work to my own children at night, because she's even done some stuff for sci-fi for children, which I think is incredible work. But today you're going to be telling us something which is a little bit outside of your direct field, right?
That's true. I'm an evolutionary biologist, and as one, I'm interested in the origins and evolution of life on Earth. And while my expertise is mostly in evolution of behavior, I'm also interested in the possibilities of where else in the universe life would evolve, what conditions are needed for it, and that's sort of a connection between my childhood interests of biology and astronomy. So I also attend astronomy and planetary science conferences, and I'm interested in the chances of discovering and characterizing extrasolar planets around other stars.
So it sounds as if, Julia, you're going to be telling us something rather galactic and universul. So indeed, maybe something you can even use to tell the time in other parts of the universe. I'll leave it to you. So Julia, the floor's yours.
Imagine a planet orbiting a rapidly spinning, dense, tiny remnant star. We are talking about neutron star planets. They are amazing, but it's about the only thing we know for sure about them. Rewind to 1991. Astronomer Alex Wolszczan sees something very peculiar with the Arecibo radio telescope. There's a class of neutron stars called Millisecond Pulsars, which act as very accurate cosmic clocks. Don't imagine like this watch. Imagine something more accurate than an atomic clock. They are very regular and very fast. But a signal from 1 millisecond pulsar was off, and there was a pattern. Could it possibly be planets tugging at the star and making the signal go earlier or later toward us? Indeed they were, and these were the first discovered exoplanets ever. Nowadays, we know 6 neutron star planets in 4 systems, each wildly different. What's their story? Could a planet survive a very violent supernova explosion in which the neutron star arises? Or could it form from a material falling back onto the remnant star? Or even some more exotic processes? We don't know. And knowing more about the planets could give us a hint. Unfortunately, we only have the masses and orbits of the planet. What are they like? Are they sizzling, evaporating molten balls of metal? Or are they cold, airless rocks? Could they possess atmospheres? We don't know. The possibilities are vast, and we need observations to test them. But is it at all possible to characterize a small rock hurtling around a tiny star thousands of light years away? Well, luckily it should. And aurorae could be the key. You know the northern and southern lights near Earth's poles. This is something similar, because pulsars have freakishly strong magnetic fields and emit lots of charged particles which collide with the planets. So if the planets, like Earth, have a magnetic field and an atmosphere, and it's a big if, they could have very, very strong aurorae in both the ultraviolet and optical spectrum. So with better telescopes, we could actually see them. So the next time you wonder what the most extreme planets in the universe look like and how they originated, imagine the northern lights dancing across their skies and how they might enable one day for us to learn more about them. Thank you.
Thank you, Julia. So, if I understood correctly, these pulsars, these very dense things are spinning around very fast indeed, are emitting a radiation.
Exactly.
And if there is a planet which has a magnetic field and an atmosphere, then it can interact with that radiation and shine off light, and perhaps we could see that.
Exactly.
So that's a very exciting concept. Has anybody found any evidence of such a planet, an exoplanet which does that yet?
Well, unfortunately not. There are many expectations about observing possible aurorae on the nearest exoplanet to us, Proxima Centauri b. It orbits a red dwarf, a very tiny star, but a normal star, like our sun, not a neutron star. And it's just 4 light years away. So with the James Webb Space Telescope, which should launch in less than 1.5 years, we should be possible to observe aurorae on Proxima b, if they are there. And with even better telescopes, we should be able to observe this phenomenon on these very distant pulsar planets.
Okay, well, anybody that's an exciting, and then we've got a question for Julia. She's able to answer almost anything in the entire universe. So you can literally ask a question almost about anything. Maybe, you obviously know a lot and understand a lot about the universe. How unique do you think it is that there is a planet out there which has all these, coincidental advantages, such as its own magnetic field, the right sort of size and gravitational pull to hold an atmosphere? Do we have any idea of the probabilities?
Yes, we have, and though the variations are vast, we know that at least half of the stars in the universe, based on the observations we have in our own galaxy, have planets. Most have more than one. Actually, we now know several systems with 7 planets, which is amazing. Our own has 8. This is the maximum so far, but probably not for so long, as we are observing more and more stars and their planets. And as to the right size and magnetic field, well, it's a question whether they are at all needed for life to arise, because life as we know it needs several basic things, and the most basic one is a solvent, liquid water in this case. And then it needs a stable enough but a bit changing environment and some sources of energy like chemical reactions or heat or light from the star. So in our own system, it is possible that there might be conditions for life, even though maybe not inhabited on some places on Mars, or in the icy moons around the giant planets, like Jupiter's moon Europa or Saturn's moon Enceladus. So liquid water and sources of energy don't seem so rare in the universe or in our own system. But it's a question whether, let's say, advanced life is. And for that, we only have our experience and knowledge from Earth. So it's really difficult to have any estimate of the number of advanced life or even civilizations in the galaxy. There are attempts to estimate just that, like the Drake equation. But we don't know the values of most of the parameters. So, like for the pulsar planets, we need observations to get more accurate values to put in the equation and then see whether it's true.
Okay. Any of you here potential budding interplanetary travelers looking forward to discovering? Yes, Dasha is.
I don't have the question, but I was asked the question, or the question came over the dinner yesterday, about some very new finding, which is turning all the knowledge about space upside down. Was it you?
Yeah, we discussed about that in the back room, that they found out a planet that actually, according to some of the most used theories, shouldn't exist.
Yeah.
Because it's so huge and is rounding around a star that is relatively so small that...
Exactly. To give you a picture of that system, imagine a planet like Jupiter, a gas giant of a very similar mass, and a star which is much smaller and less massive than the Sun. It's a red dwarf, so it has about 10% of Sun's mass, and it's just a little bigger than the planet Jupiter. It's similar to our nearest star apart from the Sun, the Proxima Centauri. And this exo-Jupiter orbits this tiny red dwarf, and they are quite comparable in terms of mass. So it's a question, how did the planet originate? Did it eat all of the protoplanetary disk around the young star, or was it perhaps captured from another system? We don't know so far, but it only shows how surprising exoplanetary systems are, because just a quarter of the century ago, before we discovered the first exoplanets, we expected most planetary systems to be around stars like our Sun, maybe a bit less massive, but not extreme stellar remnants like pulsars, or not so small stars like red dwarfs. And we expected terrestrial planets like the Earth or Mars to be closer to the star, and giant gas planets to be further away. And in fact, we've seen that the diversity of the systems is amazing, because there are gas giants orbiting practically next to the star without any terrestrial planets between them. We've seen planets on orbits that are very elliptical, more akin to comets in our own system. We have seen planets without any star that have been hurled gravitationally away from their system and are now rogue planets not bound by the gravity of another star. And that's just a small slice of the metaphorical cake of the universe, which we are just discovering. And in the next years, there are new telescopes to be launched. Just next year, NASA's TESS and ESA's CHEOPS will search for more extrasolar planets, and it's expected that they will find many thousands, maybe tens of thousands planets just in the next few years. Now we know more than 3,000, but That will be an old number very soon. And the James Webb Space Telescope, which should be launched in 2019, should enable us to describe the atmospheres of these planets. Because so far with the Hubble Space Telescope, we have been able to characterize the atmospheres of many extrasolar gas giants, these exo-Jupiters. But with James Webb, we should be able to describe the exo-Earths and to see, for example, whether they have oxygen in the atmosphere, whether they have some exo-prochlorococcus or not. So that will be really exciting times.
I tell you, Julia, you should, the ESA and NASA should employ you. I think you should do a very good job. Any other questions? Good opportunity to pick Julia's brains.
If I may ask a question.
There's one here.
Yeah.
It might be a little off topic, but we know that the universe is ever expanding, and we think it is infinite. So what is it expanding into if it is infinite?
Yes, that's a good question tied to general relativity. And it's a concept that's very hard to wrap your head around. And even most physicists don't. Actually, just very recently I spoke to a physicist at the Vědeckopoularizační observatory, and he said, don't even try it. If you try to really imagine it, you'll end up in a padded cell. So, but for me, it's not so hard. I mean, maybe I belong there already. But imagine you have like a balloon, like our prochlorococcus had. And it's a sphere if we sort of make the entrance of the balloon negligible. And we can blow into the sphere and make it bigger. But, you know, it doesn't end anywhere. It's a sphere. It goes on itself. So it's just a metaphor, but it might be useful for imagining expansion of the universe, which is already infinite.
Yeah, that satisfies me in the dimension of that expanding surface, the expanding surface.
But it's harder to imagine it in 3D.
It's still expanding into something. Or is it, but isn't it the case that it's creating that something as it's going? It's actually, it's creating
really.
All these fields.
Well, to simplify it a bit, you can imagine there is an infinite space, but only a part of it is occupied. And it's the matter that arose from the Big Bang, and it expands. And as the infinite, let's say, fabric of the cosmos is expanding, the objects formed from the matter are growing ever more distant, and the distances are multiplying faster than how the speed of light travels. So with the current rate of expansion of the universe, in many, many billions of years, we won't be able to see as much as we do now, because all of the distant objects will be beyond the horizon that light can travel by the speed of light. So in a very, very distant future, it will be a universe of darkness. We won't be able to see anything apart from our own galaxy and our satellite galaxies. And it will look like a really big black void. But I mean, that's trillions of years away. And at that time, there won't be any normal stars left.
Unless, of course, the regenerative power of stem cell research leads to... Okay, Julia, on that amazing, universal, truly universal note, I'm gonna call it an end. And thank you very much indeed. It's fascinating. So, ladies and gentlemen, friends, colleagues, that brings to an end our series of talks, or maybe not, because FameLab is a bit of a rock concert of scientific conferences, and every rock concert always has an encore. So indeed, we're very lucky today to have one final encore. Our gentleman from Portugal, David, is going to come back onto the stage and is going to tell us an extra story. So please, David, come along. Can we all have a big round of applause to thank him for agreeing to give us one last pearl of a talk?
I'm going to tell you a story. The story of the first day of your life. It all started with a date between a rich girl and a poor boy. The rich girl comes from a wealthy family, and she has everything she wants. She has proteins, sugars, energy molecules, much more than she really needs. She's the egg. But why does the egg have so many things? Because it was carefully prepared for this date by your mom. Actually, your mom already had the egg before her birth, but in an immature form. And during her life, she progressively enriches these immature eggs, making a large, complete egg each month. But there is something that our rich girl doesn't have. Half of her DNA. DNA is the molecule that contains our information, like the color of our eyes and our skin or our height. And DNA is packaged in very small units called chromosomes. Usually we have 46 chromosomes, but our rich girl has only 23. And despite all her wealth, she's not able to get the other 23 chromosomes by herself. She needs someone. And so she waits. She patiently waits on the royal palace on the top of the mountain, which is the fallopian tube, for a young man to bring her what she's looking for. On the other side, we have our poor, thin boy who barely eats and really needs someone to help him. It's the sperm. The sperm knows that the egg is waiting for someone, and so he gambled everything he had on this trip. And that's why cells' energy factories, which are called mitochondria, are all concentrated on the tail of the sperm so that it can swim as fast as possible to get to the egg. But there's one problem. The sperm has a lot of competition. There can be up to 150 million sperms interested in the egg. It's like all inhabitants of Russia were looking to the same girl. But only one can win. And so, what happens next? Well, this crazy race starts, which can last up to 4 days. The winning sperm enters the egg, the chromosomes match one another, and here you are. This is the first day of your life in which you are a single cell and you are called a zygote. But do not think the worst is over. Now an equally insane journey starts, the honeymoon, in which the zygote has to grow, travel to the mother's uterus, and survive alone for about 9 days, only with the resources brought by the egg, until finally it implants in the uterus and comes home. This is a very difficult task, and it is estimated that 2 out of 3 zygotes cannot make it and end up dying. But you made it. So congratulations. Now we just have to wait 9 months, and here you are. Thank you.
Thanks, David. Thank you very much. Thank you. Well, anybody want to ask David a question about your first day of your existence, of your life? I think, David, of course, the human story I've heard told in many different ways, but that was, I think, the nicest or the best way I've ever heard the actual conception of all of us as told as you've just done now with this wealthy young girl and the poor chap who made his way in competition of 150.
It was difficult, but he made it.
I suppose competition then is really an overwhelming theme of, because earlier you were talking about the competition within the DNA molecule for different genes, and then that is extrapolated to different chromosomes and then different cells, which can lead to cancers. And indeed, it all begins with this competition of 150 million sperm. Only one can make it. So, yeah. Well, any last opportunity to ask a question? In that case, David, I'm going to thank you very much for making that encore and that fabulous story to end what's been a wonderful morning. Thank you very much.
Thank you very much.
Well, lunchtime awaits all of us, and I'm sure you're all very hungry. Thank you very much for coming today. I hope you've enjoyed it. I hope it's been an inspiration to you all. I hope it sort of set little pulses, little impulses and neurons flickering into action that is going to lead you to become a bit more interested in any of these topics we've covered today. A big thanks to all our speakers for making the journey to Prague and really entertaining us with some wonderful new concepts, and if they weren't new, then a new way to talk about them, which has been very enlightening for me. So, and a big thank you to the organisers, Dasha in particular. A big thank you to you as well for coming along and giving us a nice introduction and a talk. I hope you've all enjoyed it. Have a nice remainder of the day and enjoy Týden vědy techniky next week. Bye-bye.